Hi student, in this video you are going to learn about lipid, and most likely in your daily life you will find lipid as in form of oil or fat. The main characters of lipid, they are mostly or entirely hydrophobic. So if you find an organic molecule that is hydrophobic, most likely it will be a lipid. Now, the other difference between lipid and our carbohydrate and protein, there is no true monomers in lipid. So what I'm saying by true monomers, the true monomers should be building blocks that are combined in repetitive ways. If you still remember, in carbohydrate, we have learned that glucose can be repeated many times to become starch and glycogen. However, in lipid, you will see later on that bigger molecule or kind like polymer are just built by some or a few number of monomers, and they cannot be repeated. We will see about this in the next slide. Now, as an introduction, there are three types of big molecules, so it's kind of similar to polymer in lipid. The three types of lipid are: 1. Triglyceride. The example of triglycerides are fats and oil. So cooking oil, the lard, and so on, they must be triglycerides. The monomers, again, this is not true monomers, you will see in the next slide, but the building blocks or monomers are fatty acids and glycerol. And the second type is phospholipid. Now, this sounds familiar for you, right? Yes, because you have learned about this in chapter 1 when we talk about cell membrane. So phospholipid is the main constituent of cell membrane. And the monomers, the building blocks, are fatty acid, glycerol, and phosphate group. We will talk about it again further in the next slides. And the last example, the last type of the lipid is called steroids. Now, you may have guessed, yes, it has something to do with hormones. And these steroids consist of 4 fused ring molecules. And these ring molecules cannot be considered as monomers because by themselves alone, they do not act or do not have characteristic as lipid. It is only when they combine they become lipid. Now, the examples of steroids are cholesterol. You also have learned about this in chapter 1, plasma membrane, if you still remember in Andrew Ellett. So the structure of the cholesterol looks like this, right? So it is a representative of ring molecules. And hormones, for sure, that for example the hormones are estrogen, progesterone, and testosterone. Let's start with the first type of lipid, which is triglyceride. The tri talk about the number of 3, which is number of fatty acid that will combine with a glycerol. So triglyceride. So it consists of 1 glycerol and 3 fatty acid. It will create a triglyceride or a fat or oil. Right? Now, the syllabus asks you to be able to draw the structure of glycerol and fatty acid. Now, it's very easy actually for glycerol. It's kind of like a zipper, right? The backbone, the main backbone, is contains of carbon, all right? While on one side it has all hydrogen and the other side all OH or hydroxyl. And then it will have an N. Both ends consist of hydrogen. So that's glycerol. And then how about fatty acid? Again, it looks like a zipper, but this time the backbone, which is carbon, the same with glycerol, it can be very long. We will talk about types of fatty acid later on in the next slide. Now, it is, and both sides have hydrogen, and also the other side also have hydrogen. It has an N, which is hydrogen as well. Now, however, one thing that you have to remember is the carboxyl or COOH. So, first the carbon will make the double bond with the oxygen, as you can see from here. And then the second one, it will make a single bond with hydroxyl OH. So, COOH. Okay, that's what you have to remember about fatty acid. All right, so again, triglyceride is made of 1 glycerol and 3 fatty acid. And they combine, of course, you have learned in the previous videos that the combination of the building blocks or monomers to become polymer always involves the condensation reaction, which means that there is one water molecule that will be produced for every bonding that is created. Now you can see that the glycerol have 3 hydroxyl. 1, 2, 3. Right? 1, 2, 3. And then this will combine with the hydrogen atom from the hydroxyl of fatty acids. So you can see that one glycerol can make bonding up to maximum 3 fatty acids. So you can see from here, they combine, combine, combine. And this OH and H will become 3 different water molecules. Alright, now as a result, it's going to be triglyceride. So you can see from here, this is not repetitive because there is a maximum number. You cannot keep on adding monomers to become lipid polymer. The glycerol can only add up, can only combine with maximum 3 fatty acid. Alright, so again, you can see here the bonding happens in the bridge is in form of oxygen. Alright, now there are many types of fatty acids. Now I can give you the examples from here. For example, lauric acid, you can find it in coconut, mother's milk, butter, and so on. And then myristic acid, you can see also in butter and coconut. And then in peanut, you have arachidic acid. Okay, and the acidity, why the name is fatty acid? Now those who do chemistry, maybe you know that this COOH carboxyl group is something that causes the acidity like vinegar. It is also have COOH or carboxyl group. Now let's talk about the second type of lipid, which is phospholipid. Now you have learned about this in chapter 1, but now let's talk about the molecular structure of phospholipid in detail. The building blocks or the monomer of phospholipid consists of glycerol, fatty acid, and this is what makes the phospholipid has hydrophobic and hydrophilic. So the head is hydrophilic, loves water, and this one hates water. So the love of water comes from the phosphate group. Now, so now you know if it is just glycerol and 3 fatty acids, all of them are hydrophobic. However, in phospholipid, the tail, now you know why there are 2 tails drawn in phospholipid. It comes from the 2 fatty acid that it will combine, okay, like in this picture. While the other one, the other OH here, will combine with phosphate group. So the representative looks like this, all right? So this is the phosphate group, and this is 2 fatty acid, and in between is the glycerol, as you can see in this picture. This is the glycerol, this is the phosphate group, and these are 2 fatty acid, all right? So again, for sure, they combine through the condensation reaction, which is not, drawn here, but this is the bridge. This is the bridge. It's similar story with the triglyceride. It is through the condensation reaction, and it has hydrophilic region, which is the head, and then the tail. So now this is the last type of lipid, which is steroids, and as it has been mentioned before, it's made of 4 fused ring molecules, right? However, each ring is not called as monomer, since by itself each ring doesn't show features of steroid or lipid. Thus, steroids do not have monomers. So fortunately, you don't have to memorize this structure. Yeah, of course, it is too hard for you, but just for your information, the steroids must consist of ring structure like this, right? And as I have mentioned before, we will learn in details about the types of fatty acid. And once again, in one of the past exam, there is a question for the students in 2016, if I'm not mistaken. Students have to draw the fatty acid, all right? Saturated and unsaturated. So again, make sure that you know how to draw the fatty acid. Let's start. In general, there are 2 classifications of fatty acid, which is saturated and unsaturated. What is saturated fatty acid? Saturated fatty acid means all of the carbon in the chain are connected by single covalent bond, like what you have seen before in the previous slide. So it's kind of like a zipper, right? Carbon C from beginning until the end, right? And all the carbon are connected with single bond. So the number of the hydrogen connected to the carbon cannot be increased. So they have been fixed. They are very stable. However, in unsaturated fatty acid, there is one or more double bonds between carbon atom in the chain. So you can see, yep, it's over here. So the rest of the carbon are combined with one single bond, while the carbon over here, they are connected to with double bond. So it means that the number of hydrogen connected to the carbon can be increased. So if there is a reaction that separate this double bond to become a single bond, this carbon can have more hydrogen, can be replaced. So the double bond can be replaced by a single bond. So it means that this unsaturated is has less number of hydrogen compared to the saturated one. And then the unsaturated fatty acid itself consists of many different types. So again, the classification consists of fatty acid, okay, divided by two, which is saturated and unsaturated. Now the unsaturated will be also divided into many different types. All right, ready? The first one. If it is only one double bond, it's going to be called monounsaturated fatty acid. All right, only one double bond. However, if it has more than 1, 2, 3, and so on, it will be called polyunsaturated fatty acid when there are two or more double bonds. And then for monounsaturated fatty acid, it will consist also two different types. The first one is cis unsaturated fatty acid. You can see this is the cis. The second one is the trans. From its name and from the picture, can you guess what is the difference? Yeah, for cis unsaturated, both hydrogen located in the same space, okay, in the same side of the double bond. However, the trans unsaturated fatty acid, the hydrogen are the opposite. Now, you will see that this classification will be useful when we talk about the risk of consuming different type of fatty acid in the next slide. All right. So again, the unsaturated fatty acid can be divided into two, which is the mono and the polyunsaturated, and the mono can be cis or trans, right? So what I have mentioned before, that knowing the classification of a different type of fatty acid will be so helpful for you to evaluate the risk of consuming different type of fatty acid. So let's talk about the healthiest one. The healthiest one is unsaturated fatty acid. And the more double bond exists in the fatty acid, the healthier the oil will be. So the healthiest oil is the polyunsaturated. You can get it by consuming corn oil, soybean oil, sunflower oil, and cold water fish. Well, this is not mentioned like seed, like of course like sunflower seeds, soybean seeds, and so on, right? So salmon, for sure, this is very healthy, and it will give you polyunsaturated fatty acid like omega-6, omega-3, very, very good for your health. And then one level lower than that is monounsaturated. This is also healthy, but not as healthy as polyunsaturated. And you can get it from the olive oil, canola oil, peanut oil, sesame oil, avocado. So those who love avocado juice, do not feel guilty because of enjoying the smoothness, the taste of the avocado that is wonderful, because the oil that exists in the avocado is actually healthy oil. So keep on eating the avocado juice, right? Okay, and then the next one is unsaturated fat. Well, it is not truly a no-no, but if you can, please limit the consumption of this fat, right? So for sure, you know that animal fats are saturated fat. So it comes from the chicken, poultry, fatty meat, butter, ghee, dairy products, full cream milk. It must consist lots of saturated fatty acid. And some vegetables also have fatty acid that is saturated, such as coconut oil, palm oil. So in daily life, you know that the cheapest oil are actually palm oil. Now, while the corn oil, olive oil, usually they are the expensive type of oil in the supermarket. Yes, it is because not only the production that is higher in cost, but also it is because simply the unsaturated is healthier, right? So creamer, so even though you put creamer in your coffee, actually is also a saturated fat. So putting milk and the creamer, you will both get the saturated, both will give you saturated fat. Okay, condensed milk also have the saturated fatty acid. Right? Again, try to limit as much as you can the consumption of saturated fat. So what is the worst one? This is what we call trans fat. If you still remember, the trans means that the hydrogen of the double bond located in the different or opposite side. So most likely this kind of trans fat does not exist naturally. They are created, they are manufactured. Right, so if you go to a supermarket, try to read a part of like the nutritional fact of biscuits or something. You will find what we call hydrogenated vegetable oil. So these oils are actually manufactured to become trans. Why the manufacturer have to make the trans fat? Because trans fat tend to be more stable in high temperature. So it's kind of like if you go to the fast food, once I experienced that. So in the morning I went to a fast food that opened for 24 hours. So in the morning they prepare for the french fries, and I saw that the oil is not cooking oil in liquid, but it is in form of like a fat, and tend to be it's trans fat that is more stable. After many times of frying, deep frying, they still can be used because they are very stable. But it is so stable so that in your body it's also stable and can create clog in your blood vessels. So it consists mostly in fast food, cakes, pastries, chocolate, deep fried food, processed food, and so on. So again, try not to consume this as much as you can, because this is the unhealthiest one. So which one then? Maybe you ask like, which one is worse? Yes, I think I have mentioned. Trans fat is worse than saturated. So trans fat are artificially made to become stable in high temperature, and this is what makes it like it is not just some people say the rumors about it. No, in patients who died from CHD, which is coronary heart disease, which means that there is a coronary artery, and the function of this artery is directly take some blood, fresh blood that is pumped out by the heart to be given to the muscle of the heart, so that the muscle of the heart will get the freshest oxygen. However, this coronary heart artery can be blocked by lipid, okay? And then this will cause what we call CHD, coronary heart disease, because when the muscle of the heart do not get enough oxygen supply and nutrient supply, of course they will die very soon, and this is what we call the heart attack. All right. Now, again, these arteries, the coronary arteries, can be narrowed by the plug, and it is found that the trans fat layer is found in high amount in people who died from CHD. After they died, the doctor do the autopsy, and they found this. So it can be said it is a causal link. It is not correlation, it's causal. However, when we talk about saturated fat, well, it is natural. Unlike the trans fat that is artificially, it is natural and mostly found in animals. And you know that people who eat too much animal fat and so on, they tend to have, again, the risk of CHD is higher. So there is a positive correlation. However, this why it cannot be causal link, because you can see in this case, for example, this is Maasai people that live in Kenya. So they cannot really have too much vegetables in their diet. So most of the time they eat animal with its fat, saturated fat. But there is no case of CHD happen there. So again, there is a positive correlation. However, there is another factor that causes CHD in people who intake too much saturated fatty acid. For example, like lack of aging. But perhaps for Maasai people here, their ancestors have adapted very, very well in this area for many, many years, okay, thousands of years. So their body can metabolize saturated fatty acid easily and do not cause any CHD. All right? So you have learned in the previous video about carbohydrates such as starch and glycogen. They are food storage. Now lipid, for sure, also food storage. So what's the difference? Why there are two types of food storage? So let me try to explain the difference so that you will know and understand the function of both. So both are important because they have different functions, right? So let's see the first one. Carbohydrate, it is a short-term energy storage. So that's why it is stored in the muscle. And so whenever you know, like between breakfast and lunch, you have in our school at least, let's say, almost about 5 hours. And if you're not lucky enough to get your break time, it means that you just have to survive with the sugar that you had in your breakfast. So it means that if you have quite a lot of breakfast, the extra sugar will be stored as glycogen in your muscle, and then that glycogen can be easily break down between those two time of breakfast and lunch to be used for your cell respiration. However, for lipid, it is a long-term energy storage. Right, so it takes more time for the lipid to be broken down. However, again, the function is not as a fast cash. It is something like you need time for the future. So if you have a prolonged time of diet, of low calories diet, or because of fasting and so on, so some people may notice like, oh, you are now slimmer. Now it means that your long-term energy storage in form of lipid has been used. All right. And carbohydrate, also glycogen, has lower content of energy per 1 gram. So let's say if it is 100 calorie per gram, it means that lipid can have content higher, almost double. So for example, it's about 200 calorie per gram. So it means that the same gram of carbohydrate, 1 gram of carbohydrate and 1 gram of lipid will give double amount of energy compared to carbohydrate. So it's a long term, right? And then each gram of glycogen is associated with 2 grams of water. So every time you want to keep glycogen in your cell, let's say in your tissue muscle, 1 gram of glycogen will require 2 grams of water to be stored. All right, so in total, your muscle will keep 3 grams of mass. However, the lipid, as you know, it is hydrophobic, so it is not associated with water. If your tissue carry 1 gram of lipid, it is just 1 gram. It doesn't have to combine with any water molecules. So, therefore, lipid will be 6 times lighter in mass to keep the same amount of energy compared to carbohydrate. 6 times. Can you calculate it using the information that I give you? Once again, the energy is double. Every 1 gram of glycogen, there are 2 grams of water. So it means that for the same amount of energy, lipid will be 6 times lighter. Can you figure out the calculation? If you can't, it means that in the meeting, in the class, I'll talk about it. But try to calculate it by yourself first, right? Well, let's continue. So efficiency in body mass is very important, okay? Especially for volant animal like bird and bat that can fly. Well, if they have such a big, huge mass, of course, it's going to be difficult for them to fly. So that's why storing fat in their body is more efficient than storing glycogen. So in daily life, we can call that glycogen is kind of like a fast cash, the money that you have in your wallet. However, for lipid, it's kind of like let's say your deposit in a bank, you know, like you have lots of money there, but of course you have to go to the bank and then do some paperwork before you can get the cash from the deposit that you have in the bank, right? I hope you understand about this. Yeah. Alright, in your classroom you will do station 4, which is the practical work, and this is gonna be fun, right? Okay, so the fat storage will be accumulated, and we can measure the health level of a person based on, you know, like the mass that you have in your body, and this is what we call the BMI, body mass index. Okay, you will be measuring your height and also measuring your weight, and there is a certain formula, as you can see in your Andrew Elledge book, page 23. Okay, if you want to read it before the experiment, please do so that when you go to the class you will do this for yourself. Right? See you.